Hi, Assalamualaikum and salam sejahtera. So my name is Muhammad Akif Irwan bin Muhammad Azani. You guys can call me Iwan. And my metric number is S55471. And today I'm gonna talk about diversity of marine phototroph. Let's check it out. So before we go any deeper, I would like to introduce you to the phototroph. So, what is phototroph? So, as you know that phototroph is the organism that can grow using light as an energy source, and then there are also photosynthetic organisms that can perform photosynthesis. What is primary producer? So primary producer is actually organism that synthesis organic compound using inorganic carbon as their carbon source. Phytoplankton serve as the primary producer in the marine ecosystem. So, did you guys think how researchers and scientists collect this plankton and bring them to the lab and study their characteristics? So actually, there are few instruments to collect them as such example, Bonko planktonate, Kitahara planktonate, Nopet planktonate. So here's the okay. This is timeline of the from traditional to new understanding of marine phototroph is begin from 1970 until 2011. So from 1917, scientists state that diatom and dinoflagellate as a primary producer, and then new species has been found in that years, which is unicellular cyanobacteria chlorococcus. So move to the 1918, chlorophyll as a oxygenic photosynthetic as a primary producer, and move to the 2002 bacteria as a primary producer, which is EABP and ANBP, which is aerobic anosogenic photosynthetic bacteria, and then for ANBP, which is anaerobic anosogenic photosynthetic bacteria, has been reported in that years. So move to the 2010 discovery of pico-eukaryotic phytoplankton, and then 2011 strange phototrophic microorganism has been found. So let's start to the 1917 to any deeper to study them. How traditional understand about primary producer. That day, plankton has been found using DNA detection and flow cytometry, and then they found predominant phytoplankton in marine environment. They state that this phytoplankton, which is smaller than collected by planktonet. What I conclude in this, haptophyte include chlorococcolithophore phytoplankton deposit calcareous plate, which is coccolith. Scientists H.M. Lility state that two microalgae considered to be major primary producer using phytoplankton detection at that time, so which are diatom and dinoflagellate, and not include phytoplankton bloom, which are red bloom and cyanobacteria bloom. These microalgae, did you know guys about their size? So actually its size is all about less than 50 micrometer. Planktonet by 10 micrometer to collect them has been used for plankton research. So net observation technique by fluorescence microscopy and accumulation from electron microscopy reveal that unicellular cyanobacteria cyanochlorococcus species has been found play a role as a, so actually this bacteria play a role significant as a primary producer. So let's start to the 1918. So scientists found chlorophyll as a perform oxygenic photosynthesis as a primary producer. There is a new development of technique. They use high performance liquid chromatography, which called CPLC, brain pigment analysis, to the study phytoplankton and brought new insight in marine picocyanobacteria. Cyanochlorococcus has been found in years. This bacteria recognized contain divinyl chlorophyll A, which is DV chlorophyll A. Absorption spectrum slightly different from using chlorophyll A, just normal chlorophyll A. They not contain picobilipigment. This is an example of cyanochlorococcus, and then these are picobilipigment. Pretty right? Cyanochlorococcus, they also found cyanobacterium which is genus for Fluorococcus. So actually Fluorococcus is the coccoid to episoida unicellular cell, and they also perform oxygenic photosynthesis using divinyl chlorophyll A or B. So as I mentioned before, Cyanochlorococcus just using divinyl chlorophyll A, but Fluorococcus using divinyl chlorophyll A and B. So they also absorb blue light and dominating to the ocean color. That time, Fluorococcus plays significant role as a primary producer. 2000, scientists discovered of ubiquitous photoheterotroph, actually first recorded in 1979. They found two significant that microorganisms contribute to the transformation of light energy into chemical energy in marine environment. They found aerobic anoxygenic photosynthetic bacteria and anaerobic anoxygenic photosynthetic bacteria. They actually photoheterotroph without carbon fixation and never recognized as a primary producer. You guys think why? Anaerobic anoxygenic photosynthetic bacteria is a non-sulfur purple bacteria. Here's other example of bacteria. And then another one, anaerobic anoxygenic photosynthetic bacteria, which are alpha proteobacteria. And then this is the picture. How pretty, damn right. Actually, alpha proteobacteria is the perform photosynthesis without oxygen. AABP, a non-sulfur purple bacteria, use oxygen, but alpha proteobacteria perform photosynthesis without using oxygen. You guys know what? Alpha proteobacteria use bacteriochlorophyll instead of chlorophyll. New technique discovered from Sunday. Scientists found proteorhodopsin was detected. Actually, rhodopsin is like protein. Here's the example of rhodopsin. And then this the link you more detail. Rhodopsin is contribute to ATP synthesis. It has halobacteria in archive transform light energy in transmembrane proton electrochemical gradient. Actually was detected in large amount in seawater by metagenomic analysis. What is metagenomic analysis? Metagenomic analysis involves application of bioinformatic tool to study the genetic material from environmental uncultured microorganism. So this is the approach to perform metagenomic analysis. This is the picture of the rhodopsin. New technique discovered, which is far red light. Far red light using to detect oxygenic photosynthesis. Chlorophyll A is defined from light photosynthetically active radiation. Part for evolution of light strength for activity of oxygenic photosynthesis. Far red light found three chlorophyll, which is chlorophyll A, chlorophyll D, chlorophyll F. Chlorophyll D used by cyanobacterium, which is Acarichlorosis marina, to perform photosynthesis. This bacteria absorb far red light for 30 nanometer longer wavelength than chlorophyll A. The chlorophyll S was detected from cyanobacteria isolated from microbial mud. This all thing require consideration of the part concept and primary production in marine environment. We move to 2010. So discovery of pico-eukaryotic phytoplankton. So eukaryotic phytoplankton was revealed in significant amount in marine environment. Scientists found three phyta: chlorophyta, heterocontophyta, haptophyta. Using metagenomic analysis and flow cytometry technique that I have mentioned before, show that significant amount of pico-haptophyte. They found undescribed species. Undescribed species were detected by 19 HF. Amount of microalgae in unknown phylum was reported in that time. Found DNA sequence close to cytokine hinage. Just the example of cytokine hinage. But phytoplankton in tropical and subtropical do not contain 19 HF specific to the algae in Haptophyta, because they specific to the algae in Haptophyta. Here's the example of phyta, heterocontophyta and Haptophyta. Pretty right. The last year's discovery of traditional to new understanding of marine primary producer, 2011. Using metagenomic analysis in 2011, they found new phylum was detected targeting plastic DNA. Another one is Cyanobacterium light particle UCYA of nitrogen fixing in marine water. They found nitrogenous gene in the cyanobacterium like protein. And this is of the UCYNA. Although they find Phototrops as an organism synthesis ATP using light acid energy source. In other words, Phototrops are organism synthesis ATP by ATP synthase. Can you all see how important and great is the diversity of marine Phototrops for all of us? Marine life benefit many people around the worldwide. So for example, in medicine, community and others, therefore we must study and understanding about marine life by understanding their food web and energy flow. That's all from me. I hope you enjoy my presentation. Salam COVID-19 and thank you.